Hello, my name is Alejandra Tristan, and I will be doing my overview on melatonin. Um, my partner and I chose to study melatonin because of how it helps one sleep. Although melatonin is a hormone um, produced naturally in the body, some people don't, produce high levels of it or enough to naturally induce sleep. And that's why I chose to do melatonin because I have to take melatonin to help me sleep. Um, Melatonin is naturally occurring, it's a hormone with no record of toxicity. However, it can be used therapeutically um, and can be chemically synthesized and it's chemically synthesized from the pineal gland. The pineal synthesis and secretion of melatonin is dependent on the a receptor function, and melatonin in humans is involved in the regulation of many biological functions, including sleep, circadian rhythm, the stress response, aging, immunity, and reproduction. Um, we go through the synthesis in our paper, but basically it's a multi-step synthesis, and it starts with tryptophan and ends with obviously melatonin. It goes from tryptophan to 5-hydroxytryptophan to 5-hydroxytryptamine, which is serotonin, and then N-acetylserotonin to eventually melatonin. And the basic biochem of our drug of choice, melatonin, is that melatonin is produced by the pineal gland in response to darkness. The hormone is not stored in the gland, however, it is secreted into the bloodstream and can penetrate any and all parts of the body. The nighttime synthesis and release of melatonin is controlled by the SCN clock, known as the suprachiasmatic nucleus clock, and obstructed by light conditions. In humans, detection of light makes activity in the retinal ganglion cell that that projects to the CSN in the hypothalamus, causing the release of the inhibitory Y-aminobutyric acid that subdues the circuit regulating melatonin synthesis and the release. The synthesis of melatonin on the the blocking. So the MT3 receptor is was found to be basically, or discovered to be the enzyme quinone reductase 2, which is what we show or what we use to show in our ligand interaction between our chemical or our drug and the enzyme quinone reductase 2. So the biological interaction that we chose is the one I just mentioned. And we're going to go into that right now. Let me share my screen. And here it is. We are on the RCSB protein database for our drug, melatonin. We're going to go down to, oh, here it is, 2QX6, crystal structure of quinone reductase 2. We're going to go down to the small molecule. And here is basically, you know, the closest thing to melatonin. It's just iodo. Here's a ligand interaction. Click on that. And then here we have our drug or our molecule, I guess. We can take off the polymer. We can remove the water. And there you have it. It's right here. All right, so that is our ligand interaction. And basically we can see in our interaction, we can see hydrogen bonds, disulfide bonds holding the protein to the molecules, the interactions. We have London dispersion forces as well. But here would be our molecule, and then we can isolate it by just removing all the other stuff. And there it is. All right, thank you so much.
Hi, I'm Valeria. I'm Alejandra's partner. So a little background on melatonin. Melatonin is produced by the pineal gland in response to darkness. And it's not stored in the pineal gland, but it's secreted into the bloodstream, and it can penetrate like all parts of the body. So it's released by the SCN clock, known as a suprachiasmatic nucleus clock. And obviously, like, it's obstructed by lighting conditions. So it's released like when it's dark, basically. And so right here, when I pull it up in Chimera, we see our molecules. So here, we see the enzyme 2QX6 interacting with the crystal structure of quinone reductase 2. So this is actually also MT3, which is a binding site of melatonin. And actually, melatonin inhibits QR2, and it acts as a binding site. However, we don't know how it inhibits, like the mode of inhibition. Yeah, so we don't really know the mode of inhibition. We just know that it does inhibit the enzyme. There's still like a lot of research being done, as it's pretty recent that it's been discovered to be like a binding site of melatonin. So here, if we get rid of like the chains and the ribbons, right here we can see it closer, and so we can see that interaction up close, and we can see right here are the hydrogen bonds. So obviously there's London dispersion forces going on. There is also dipole-dipole interactions, as you can see right here, and there's also cation pi interactions going on as well. And it is an FAD linked enzyme. So it's actually, FAD stands for flavin adenine dinucleotide. So right here. So right here, you can see all of this is the FAD. And yeah, this is what it looks like right here.